So in today's lecture, we're going to start talking about the working cell. And the working cell is going to be all about how we can take all of those different organelles that we've talked about previously, have them come together, perform all of their jobs, and ultimately create kind of this living cell. And when we get to multicellular organisms like ourselves, all of those individual cells are going to have specific jobs to do. And we'll talk about the different processes that are going to be going on within those cells. So you can think of the cell as a machine, and the machine of the cell is going to require movement, energy processing, and production of different products. So for example, making proteins. Cells are going to need to control their chemical environment. So they want to maintain an internal environment that's separate from the external environment. In order to do this, they're going to use things like energy. So they're going to have to use energy to make sure that they're moving different molecules from one side of the membrane to the other. They're going to do lots of chemical reactions with these proteins called enzymes, and we'll talk a lot about enzymes today. And they're also going to control the plasma membrane by allowing certain molecules to come in and out. Okay, and so that plasma membrane is this barrier protecting the inside of the cell from the outside of the cell, and it's going to be important to maintain that barrier and to regulate what can go across that barrier. In order for the cell to perform the different functions it has to do, it's going to require energy. And so let's talk a little bit about what energy is. So energy is going to make the world go round. It is going to allow for us to be living and life on Earth. It's going to allow us to ultimately power all of our vehicles and everything that, it's really hot right now, so everyone's probably with their air conditioner on if you have one. That requires energy, okay? And so energy is essential for life and all of these processes we're going to talk about today. But what is energy? So ultimately, we're going to talk about the sun, because that's where our initial source of energy is going to come from. And we'll talk about how that sunlight energy actually is used in our bodies. But let's get some basics about energy first. So energy is going to be defined as the capacity to perform work. Okay, so work is, for example, moving your muscles, okay, or breathing, your heart pumping your blood, turning on a light, having a fan turn around, okay? All of that has energy and it's performing some type of work. So instead of just being, a rock doing nothing, we're going to, if you want to have that rock have some energy, you take that rock and you pick it up and then you can throw it and you can actually create some energy with that rock. Okay, so kinetic energy is going to be within the total kind of umbrella of energy, and it's going to be the energy of motion. So for example, if we throw a rock, that's going to have energy in it because we are putting motion into that rock. Potential energy is stored energy. And so you can think of that, if we pick up a rock and we kind of cock our arm ready to throw, that's potential energy. So we haven't released the energy yet, but it's stored, ready to go. So let's talk a little bit more about potential and kinetic energy. So in this example, which is very similar to what I was explaining before with the rock, so if we look at the brick by itself, the brick does not have any potential energy. But if this man picks up the brick, he's actually going to allow for it to have some potential energy. And this brick is going to fall, and while it falls, that will be kinetic energy. But the energy is actually coming from the gravitational pull on this object. So the reason why everything falls to the ground when you drop it is because of gravity. So if this man picks up the brick, he's doing work, first of all, to put the energy in that, and he then has some potential energy stored by holding that brick. The second he lets go, this is going to be kind of movement, which is then going to be kinetic energy. Now this is very similar to what you would have if you have a slingshot or if you just have a rubber band. So if we're in this first stage, you have kind of the slingshot with some pebble in it. There's not really any energy in this, but if you pull back on the slingshot, you're storing in energy, and this is, for example, elastic energy. Okay, this is the stored potential energy, and the second you let go, it's the energy of motion, which is going to be kinetic energy. So things can go from one form to the other, potential to kinetic, over and over again. Okay, and we're going to start talking about kind of conversions of energy. So when we look at these different conversions, both machines can make these conversions, so your car can do this, but also organisms will do this as well, where we're going to transform kinetic energy to potential energy and go back and forth constantly. Now, in these energy conversions and transformations, energy, the total amount of energy is conserved, okay? We cannot create or destroy energy. So that is kind of one of the law of physics. You cannot create new energy and you can't destroy it. It's conserved. You can just transform it into different forms. Now, this is called the principle of conservation of energy and is important in all different facets of life. Okay, and so we conserve energy. We will talk a lot about how you can have some of the energy lost in heat, but it still is considered energy, and so it's conserved. So you're never kind of losing the amount of energy in total if you look at the entire system. So here are some examples of transformations of energy. For example, when you eat, so if you eat an ice cream cone and then you go ride a bike, you are converting chemical energy that's stored in the food. Your body is breaking it down and forming it into kind of ATP that can then power the muscle contractions that require you to move or ride a bike. We can take radiant energy, which is going to be solar radiation, and turn that into chemical energy. We have a whole lecture about photosynthesis, which is this exact process right here. We have chemical energy that we can put into our car, and that can cause motion. This process right here is cellular respiration. This process right here is very similar to respiration. It's just not in our bodies. And then, for example, when you plug in something like your stove or a blender, you're taking electrical energy and you're turning it into thermal energy. Okay, so these are energy transformations. And the main thing to understand is that what comes in is going to be the same that comes out. So the total amount of energy is staying the same. Here's a couple more examples that just give you a little more detail. So for example, when you hammer a nail, it changes mechanical energy to deform and heat energy. So if you've ever hammered something and you feel the nail right after you've hammered it, it often is going to be really hot. And so part of the energy that is happening in that transformation is given off as heat, and you can actually feel it. So before we move on, I want to talk about entropy. And when we talk about energy conversions, there always is going to be a part of the energy that is not usable in many cases, and it's lost as heat. Now, when I say it's lost, it doesn't mean that it's gone or it's destroyed. It's just in a different form that we can't necessarily use actively to power something. And every energy conversion will release this randomized energy in the form of heat. So that's just standard. Heat is going to be random motion of atoms and molecules. So when we talk about molecules moving across a membrane, one of the reasons why they move is because they have this random motion. Okay, and we'll talk about that in a little more detail. Now, this is a type of kinetic energy, because if something's moving, that's going to be kinetic. Okay? And during every energy conversion, there's going to be some heat that is produced. So this is just kind of something that always happens. Now, most of the energy from heat is going to be in its most chaotic form. So it's aimless molecular movement. So it's just, again, random, so molecules are moving. And I bring this up, it is kind of a difficult topic, but it's just interesting to understand what's happening, because we will talk about both cellular respiration, photosynthesis, and some of the byproducts of those is heat being produced, okay? And it's important to understand where that heat's coming from. Well, one of the reasons why our body temperature is 98.6 degrees Fahrenheit is because we do all of these chemical reactions, and one of the chemical reaction byproducts is heat, which helps us keep our temperature. Now, entropy is kind of a measure of disorder. So if heat gives off and it has this random motion, aimless. So what tends to happen as you do energy conversions is you increase the chaotic or disordered randomness of the universe. So that's what this image is trying to kind of show, where you start off with the word entropy. You can see the letters pretty clearly, but heat, which is going to be kind of this random aimless motion, is going to start spreading out and increase this entropy of the universe, or this randomness or disorder. Okay, so we're now going to start talking a little bit more about chemical energy and how it relates to life. So molecules are going to store energy in their arrangements of their atoms. So when we talked about those chemical bonds, ionic, covalent, and hydrogen, there is going to be some energy stored within those bonds. Okay? Many organic compounds are going to have high levels of chemical energy. So for example, carbohydrates or fats are going to have really high amounts of energy stored in their chemical bonds. Okay? You can think of a cell as very similar to an automobile engine. It's going to work in a very similar way, and it's going to be the breaking down of these larger chemical components, and the bonds, when you break them, are going to release energy, and they can allow for work to take place. In this little image right here, we have what's going to be what we talk about for photosynthesis, where sunlight comes in. Inside the plant cell, we're going to take some just basic ingredients of carbon dioxide and water. The light is then going to help convert these from solar energy into chemical energy, which is carbohydrates, and we also release oxygen. So we'll talk about photosynthesis in great detail. So here's how a car and a cell are very similar. The only difference is what we're actually breaking down. So for a car, it's going to use gasoline and oxygen. So you have to have oxygen for this to take place. They're going to have combustion take place in the engine. Part of the byproduct of that is that you're going to move your car, but it also is going to get hot. So if you've ever felt a car that's just been driving, the hood is often going to be pretty hot, and you can't touch any of it, especially if you're opening up the hood and going down into where the engine is. It's extremely hot because a lot of energy in the form of heat is given off. But what we're using is this kinetic energy to move our car. So that's the energy conversion, okay? The byproducts of this are carbon dioxide and water. Okay, carbon dioxide, which is going to be, a normal molecule that we have, that we even produce as a byproduct. Unfortunately, cars are going to produce it at much higher rates than normally. And so this is one of the contributing factors of climate change. If we go down to our cells, we would take something like food, and this specifically looks like a fat, triglyceride. So it has those fatty acid tails, and those tails, those carbon-hydrogen bonds, you can break them up and use it to perform cellular respiration. There's going to be ATP that's produced, and ATP is going to be our molecule of energy that we are going to use directly in our bodies. And we'll talk all about ATP in great depth when we go over both photosynthesis and cellular respiration. Now, this isn't a conversion that is going to create complete, all of the energy converts into ATP. Some of it's going to be lost as heat energy, okay? And the byproducts are going to be carbon dioxide and water. So very, very similar, just have different kind of fuels to get us making that energy, or converting the energy, I should say, since we don't make it. So cellular respiration is going to be the process where we take these energy-rich molecules like carbohydrates and lipids and break them down to then extract the energy so our cells can do different types of work. Now, when humans do this, our cells are going to convert about 40% of the energy in our food into kind of ATP that then can be used for work, such as making proteins or contracting our muscles. Now, 40% doesn't seem like a very efficient kind of conversion, because that means that 60% of it is lost in heat, but it's enough to power our cells and is good enough for us to maintain life. In this image, you see, this is basically cellular respiration all in one image. It is a complex process that we will talk about and go over how we go from, something like carbohydrates, like glucose, and turn it into ATP, which is going to be this molecule that will allow us to perform the work. When we eat food, we are going to be eating fuel, and the way that we measure the amount of energy in our food is through this term called the calorie. And a calorie should be something that most of you are aware of. It's on the back of all of our food with the nutritional information. What the calorie is telling us is how much energy is stored within the chemical bonds of our food. So 1 calorie is the amount of energy that raises the temperature of 1 gram of water by 1 degree Celsius. So that change in temperature is the energy that is stored within the chemical bonds of the food. Now, food calories are technically kilocalories, or equal to 1,000 calories. We're not really going to worry too much about this, but sometimes when you look at the back of different food packages, you'll see kilocalories versus calories. And it's just kind of a different way to tell you the same information. So here is just some categories of food and the amount of calories that are within them. And so these calories, for broccoli, you have 25 calories, or pizza, about 181. Now these food calories, we can then look at them and see, well, what kind of activities could we do for the same amount of calories? For example, if you're running, it's going to expend about 979 calories for a person that's 150 pounds for 1 hour of activity. Or dancing, or sitting, writing, listening to this right now, you're probably burning about 28 calories of energy. Okay, and so this is going to then, how are you fueling this? Well, that's going to come from the food that you eat. So we need to talk a little bit more about ATP and cellular work. So cellular energy, again, is going to be stored in the chemical bonds. So for example, we're going to talk a lot about glucose, which is this monosaccharide. The bonds hold energy, and we break those bonds through a process called cellular respiration. So we're going to break these bonds in the glucose, and through that process, and it's got multiple steps, we're going to start to form ATP. Okay, and ATP is going to be this really important energy storing molecule that we use in our bodies, and all different cells, single-celled organisms, plants, all of them use ATP in order to kind of have energy. Okay, so let's look a little bit at the structure of ATP. So ATP is going to be what we call adenosine triphosphate. This adenosine is actually going to be from DNA, so we talk about ATGC. So adenosine, that A, is going to be kind of one of the parts of the ATP. The triphosphate stands for 3 phosphates that are going to be attached to this adenosine. And this whole molecule is called adenosine triphosphate. Now, within the bonds, specifically this bond right here between these last 2 phosphates, is a lot of energy. And so when we break this off, this bond right here, it will release the energy that we can then use to power our bodies. Okay, so again, ATP consists of adenosine plus a tail of 3 phosphate groups, and the energy is going to be stored within that phosphate group at the end. When you break down and release the energy for work, you then form something called adenosine diphosphate, di meaning 1, 1, 2, or sorry, di meaning 2, 1, 2, and that is going to be kind of an uncharged state. Now this phosphate can be transferred to another molecule, and we can also recycle this and create another adenosine triphosphate, but we have to have some energy that we can put back into this molecule so that it can be used again. So when you think of ATP, I want you to think of sort of like a battery. So the battery can go from its charged state, which is ATP, to an uncharged state. And all of it has to do with kind of that last phosphate being attached or not. So it acts as an energy shuttle, and it's going to store energy it obtains from food in that bond for the ATP. When that bond is broken, and we take that last phosphate group off, it's going to give off energy, okay? And then we can use that ADP and form again another charged battery, or the ATP form, by breaking down more food energy. Now, the phosphate transfer is really important. So ATP will energize other molecules by transferring phosphate groups to them. This energy will help cells perform different types of work. Now, I wanted to talk a little bit about an example of this. So we have ATP, but you've probably heard of creatine. So if you're really into building muscles and kind of exercise, you may have heard of creatine. Now creatine is a molecule that can have an extra phosphate attached to it, and it will store kind of some energy in its bonds when we have something called creatine phosphate. So you can think of it as sort of like ATP, but it's a little bit of a longer term storage for that extra energy. So your cells will have a lot of creatine phosphate, specifically muscle cells, that will then be able to kind of be converted into ATP and creatine. And you can use that creatine phosphate up when you're exercising. It's kind of one of the first forms of energy that your muscles start using right away. And then you start to have to rely on cellular respiration to start breaking down more glucose to make more ATP. But that creatine phosphate has that phosphate added to it that allows for kind of storage of that energy. So when we have our cells performing mechanical work, transport work, or chemical work, they're going to need ATP. And so they'll transfer their phosphate to different molecules to allow for that to happen. So here's an example of how this happens in muscle cells. So what we're looking at right here is muscle contraction. So you're going to see that ATP can attach to this molecule right here. So you may not know much about muscle contractions, and I will not ask you about this on a test, but this is just an example of how this works. So in a muscle, we have something called actin, which is this protein structure right here, and then we have these other things called myosin. Now myosin is going to bind to the actin and contract, and you can see how it pulls the actin. That's actually causing the muscle to shorten. But the only way that this myosin can attach or actually detach is when ATP comes over and it's going to join to it, and it forms this ADP and phosphate. And so it's going to charge this myosin head that allows for contraction to take place. So this is just an example of how ATP is going to power some cellular work. So in the ATP cycle, this is going to be using the ATP. So ATP is recycled from adenosine diphosphate through the process of cellular respiration. So we have a bunch of ADP circulating around in our bodies. When we do cellular respiration, we're going to add the energy to that ADP and form ATP so that we can then use that for energy in our cells. When we have a working muscle, so this is the kind of microscopic structure of a muscle, it's going to spend and recycle about 10 million ATP molecules per second. So when we're talking about ATP being used, it's on a massive scale inside of our bodies. Okay, so lots and lots of ATP using. And this is one muscle cell. So think about, we have lots of muscle cells. So each one is using about 10 million ATP molecules per second. And it's cycling between ADP and ATP. So another thing that happens when we're talking about muscles, but we're going to start talking about the membrane. So this is the cell membrane. And I don't remember in this image if this is the inside or the outside. It doesn't really matter. But one of the things that is going to happen is that we are going to start talking about how molecules can be moved against their concentrations. So usually things move from a high concentration to a low concentration. This is generally what happens. But we can actually move molecules against their concentration gradient, so where they're higher concentrated. The only way to do that is by using actual ATP. So we'll talk about this a little bit later in this lecture about how ATP will function in this thing called active transport. We're moving molecules across our membrane from a region where they're low concentration to high concentration, which is the opposite of the natural direction of movement. Okay. Here's another example of transport proteins. So we have these proteins that sit within our cell membranes, and they will be able to move solutes, which are the dissolved molecules in our fluid, from one area to the other, but they require ATP. And so you can see how ATP is going to be involved in transporting different molecules across a membrane. And here's my funny example of this. So we have like an extracellular membrane and the cytosol, and in this case we have Drake, our protein, that's going to help expend the energy of ATP and move these molecules across the membrane, different directions. So here we just have an overview. We'll talk about cellular respiration and how cellular respiration is going to take the energy from glucose molecules and other food molecules. We'll harvest that, we'll make ATP, and then we'll use ATP for cellular work. Okay, and the energy that is stored in the ATP will be used, and we can then have ADP formed, which is then kind of the uncharged battery. We can recharge it and it can continue on in this cycle. So now we're going to move on to enzymes. Enzymes are going to be proteins that are going to allow for chemical reactions to take place. Okay, and so in here we have a piece of DNA that is surrounded by different enzymes on this image. So here's the DNA, and we have some enzymes that are going to be working on this. It could be, for example, we'll talk about how we replicate the DNA, and so this could be something called DNA polymerase, for example. But it's going to be an enzyme that allows for different chemical reactions to take place. So when we say the word metabolism, this is going to be all of the chemical reactions that are going to happen within an organism. So, if we have a bacterial metabolism, it's going to be the bacteria and all the chemical reactions that happen within that one cell. But also it could be a human, where we're talking about all the different chemical reactions that can happen within our body. Now, metabolism we tend to think of is, you can have a high or low metabolism. Sometimes people say, well, I have a really high metabolism, so I never gain weight. Or people say I have a really slow metabolism, so I gain weight easily. It's not really the same. We're not using the term necessarily correctly when people say that. It just has to do with all the chemical reactions in your body. So that would be more of a technical definition of metabolism. Okay, so almost no reaction in the body or in any cell will take place without help. They can sometimes, but chemical reactions require help for them to happen within a shorter period of time. So sometimes a chemical reaction will take days to take place, but we need it to take less than a second. Okay, and so that's going to require help in the form of an enzyme. Okay, so enzymes are going to help chemical reactions proceed. The enzymes are all going to be proteins, and they will help speed up chemical reactions. Now, one thing about enzymes is all of them have names that end in ASE. So this is amylase, for example, which is going to be an enzyme that breaks down starch into glucose molecules. Okay, so amylase, ASE at the end is going to mean this is a protein. We'll talk about DNA polymerase or lactase. So that ASE is always going to signal that this is an enzyme. So this is how enzymes work. So enzymes are what we call catalysts, so they're going to be kind of allowing for this reaction to take place. Enzymes are these proteins, and so we can see an enzyme right here in purple. The enzyme is going to bind to these different substrates, and the substrates could be different, for example, it could be a glucose and a fructose, for example. So two different molecules that are going to come in and they're going to bind to the enzyme. The enzyme will then attach to those and form this thing called an enzyme substrate complex, where you have the enzyme, which is the protein that's helping this happen, and here we have, for example, the two substrates. Something's going to happen within this molecule. So for example, in this case, we're turning it into this blue and yellow product when they start off as a red and green. So it's a rearranging of the chemical bonds within these molecules. The enzyme will allow for this to take place, and then once the chemical reaction has taken place, it will release the substrates and can go and kind of start this process all over again. So they're recyclable. So you basically can continuously use enzymes over and over again. Now, enzymes are going to help with something called activation energy. And activation energy is kind of the initial energy that is needed for this chemical reaction to proceed. Okay, so it activates these things called reactants, which are going to be the molecules that are involved in this chemical reaction. And in order to activate them, so kind of get them ready to go into the chemical reaction, they have to have some energy. Okay, and once the activation energy has happened, it will trigger the chemical reaction. And so, for example, in this reaction right here, we have this X molecule and this Y molecule, and we want to form XY. But in order for us to have this take place, we need some energy input, okay? Because it's going to need to break some bonds and manipulate these two molecules so that they're ready to bind to each other. The energy we use is going to be ATP. And this activation energy is always required for these different reactions to take place. Now, what enzymes do is they lower the amount of activation energy required for the chemical reaction. So without the enzyme, it would require more ATP than it would with an enzyme. So basically, the amount of energy required to start this reaction is going to be reduced if we have an enzyme. So this is kind of a way to explain this. So obviously this is not really a chemical reaction, but here is the situation if we had no enzyme. So we have these little frogs, which we call the reactants, and we need them to get into the water. And so they have this barrier that they have to get over, okay? And a lot of the frogs aren't going to make it over to the water. Okay, so this is going to happen at a very slow rate, so maybe it's going to take them mini jumps for them to get over. Sometimes they'll have to rest because they've jumped too many times, but it's going to take a really long time for all of these frogs to get over this barrier and into the water. What an enzyme does is it's going to reduce the barrier and lower it, which allows for the frogs to move over into the water much quicker. Okay, and so it will happen significantly faster because of the presence of this enzyme. And so what the enzyme is going to do is it's going to reduce the energy required to initiate this chemical reaction to take place. So this is what that looks like. So when we have a chemical reaction, we are going to be talking about energy. So energy is required to cause this reaction to take place. If we do not have an enzyme, the amount of energy that is required to get this chemical reaction started is going to be pretty high. Okay, so this is what happens if we have no enzyme. If we have an enzyme, what happens is we then reduce the amount of energy required for this reaction to take place. So this is the difference in the energy that is required. Okay, and so what's going to happen is that with the enzyme, we're going to have less energy required to initiate this chemical reaction, and it's going to proceed much faster than it would without that enzyme. So here is kind of an example with actual molecules. So we have in this situation A and B that are attached to each other, and C and D. So these are our reactants. And in the end, we want to have A attached to C and B attached to D. Now, the reason why we need energy is we need to put strain on these chemical bonds. Okay, so you can see how they're kind of wavy right here. They're bound to each other, and to break a bond, it requires energy. And so what's happening is that an enzyme is going to put strain on this bond. So you can kind of think of it as, you have this A and this B, and they're attached to each other. They're in your hands, and you're trying to pull them apart so you can break the bond that's between them. So that when that happens, they can then bind to something new. But you have to be able to break that bond, which requires that energy, and that's that activation energy. Once we kind of put that stress on that bond, it's going to be easier for these two molecules to then form a new bond with a new molecule. Okay, and so this would be what we have with our end products, but you need that energy to put the stress on that bond. Okay, so bonds must be broken before chemical reactions can proceed. The process requires molecules to absorb energy from their surroundings. Okay, and this is the activation energy. Okay, so it's going to activate those reactants so that they are going to be ready to break the bonds and form new ones. Now, enzymes are going to be organized in different parts of the cells because they need to be where the chemical reactions are going to take place. So certain chemical reactions will only take place in certain locations. So for example, if we're talking about cellular respiration, and that cellular respiration is going to require enzymes, that's going to take place in this organelle called the mitochondria. Okay, so we have multiple mitochondria in the cell. If we look inside the mitochondria, it's going to have specific enzymes that are only needed for cellular respiration, for example. Okay, and so this is going to make sure that the enzyme is in the right spot so that it can perform the job it needs to perform. Okay, we don't need mitochondrial enzymes outside of the mitochondria because they won't be able to function there. Enzymes are often very specific and will only do one chemical reaction, so they'll only help with one specific thing. Now, the way that we can think of reactants and enzymes is this idea of an induced fit. So enzymes are extremely selective in their reactions that they will help proceed. So catalyze just means that they're going to help them proceed. So they will have a substrate. So here's our substrate, and here's our enzyme. And you're going to see that the way that they're shaped, okay, the shape is extremely important for the function, is going to be able to fit inside the enzyme. Okay, and we call this place where the actual enzyme will bind to the substrate an active site. So the enzyme will bind to this substrate. When it actually binds, both the enzyme and the substrate will start to kind of modify their shape. So the enzyme will actually slightly change so it can bind the substrate completely. Okay, the selectivity is based on the shape of these different substrates. Okay, and so the enzyme will only be able to recognize a specific molecule based on that shape. Okay, the enzyme substrate is going to be unique and it will only fit with a unique enzyme. So shape is absolutely essential. There are certain diseases that are caused by kind of enzymes being the wrong shape. Okay, so if you have an enzyme that's supposed to attach to the substrate, but there's something wrong with the shape of it, it won't function correctly. Okay, and so these are basically going to be able to attach to each other because of their corresponding shape with each other. Now, the induced fit is this idea where when the substrates come and bind to an enzyme, they're going to change the shape slightly. So this enzyme is going to change shape slightly to better fit and bind those substrates. So you can see how just slightly these are different shapes. So specifically look at where the active site is, so where these bind to each other. They're going to change slightly. And so this is what we call induced fit. So when you have the substrates come in, they're actually going to cause the enzyme to change shape slightly so that they bind really well together. Okay. Once this is happening and we have the induced fit, the enzyme is now going to be able to allow for the chemical reaction to proceed, and it's going to start basically forming the bonds of these different chemicals, substrates, so that they can start to change and allow for that chemical reaction to take place. Okay, once the enzyme is done, kind of helping the substrate undergo a chemical reaction, it will then release these substrates and will be ready to absorb and take in new substrates. All right, so here's an actual example of how enzymes are going to work. So we have an enzyme that's called sucrase. Okay, so this is sucrase. It has the ASE at the end. It's going to have an active site, and this active site is actually going to bind something called sucrose. So sucrose is going to be a sugar. So sucrose is the substrate, and sucrose is going to be a glucose molecule and a fructose molecule. So sucrose is something that you eat a lot, so it would be table sugar. So when you're cooking, you usually use sucrose, and it's going to have a glucose and a fructose. And the whole point of this chemical reaction is to break this bond so that we have an individual glucose by itself and an individual fructose by itself. So the substrate is going to bind to the enzyme sucrase, okay? And you're going to notice that sucrase and sucrose are very similar. So this is an enzyme that breaks down sucrose. So it's going to bind to the substrate, and it's going to have that induced fit where it kind of changes shape so it can really bind well. And what's going to happen is it's going to cause this bond to break. And so what it does is this enzyme is going to put pressure and strain on the bond between the glucose and the fructose. It's going to have water come in, and it's going to do a hydrolysis reaction where it's going to break the bond between the fructose and the glucose. Okay, so it's going to help catalyze this reaction so it can take place much quicker. At the end, the enzyme will release the fructose and the glucose and is ready for a new molecule of sucrose. So it's going to continuously be able to go from, being empty, binding the substrate sucrose, then it's going to break the bonds and then release the different molecules. Okay, and so all of this would not take place at the rate that it needs to take place without this enzyme. So enzymes and substrates are going to interact on a kind of molecular level where the substrate will have corresponding chemical kind of features that are going to then bind to the enzyme. And so here is the enzyme, this big blue, has lots of different molecules, probably lots of carbons and other hydrogens and oxygens, things like that. And then the substrate, this little red thing, is going to then bind in this active site. Once it binds, the enzyme does change shape slightly, and again, this is called that induced fit. And so then it's going to be able to help this chemical reaction take place. What this chemical reaction is, I don't know. It's just, this one is just showing kind of how these are fitting together. Now, enzymes can be inhibited. So there are times when your body or in a cell, you do not want the enzymes to be functional because you don't want something to proceed. So you can inhibit an enzyme. Now, when you do this, what happens is you're basically blocking either the active sites of the enzyme, or you're having a chemical that attaches to part of the enzyme, then makes it so another molecule can't bind. Okay, and so this is going to stop an enzyme from helping the chemical reaction take place. Okay, so you can do this through blocking the active site, or you can bind at a remote site, change the shape of the enzyme, thus blocking the active site. So basically you're making it so the active site cannot bind its substrate. Now, enzyme inhibitors, they are going to prevent metabolic reactions from binding to the active site. So basically they're preventing the reaction from taking place. So here is an example of an enzyme inhibitor. So here we have our enzyme, and this is the substrate that it normally binds, and it's going to bind to the active site. And so this would be kind of our normal situation with the enzyme. So whatever chemical reaction could take place will happen. If we have an enzyme inhibitor by a substrate imposter, which means that it is able to attach to the active site, but it's not the actual substrate, and will actually block the substrate. So this is an enzyme inhibitor that blocks it so that this chemical reaction cannot proceed. Okay. Another type of inhibition is when you have an inhibitor that will bind to another part of the enzyme, so it's not blocking the active site directly, but what happens is when it binds to this enzyme, it's causing a change in the shape of the enzyme so that this substrate can no longer bind. Okay, so in this case, even though the active site is technically open, the substrate is not able to fit inside of it, and therefore the chemical reaction will not proceed. So here's an example of an enzyme inhibition. So we have this molecule that we're seeing here, which is a trypsinogen molecule. This is a precursor to a digestive protease enzyme trypsin. Okay, so trypsin is going to be an enzyme that's going to help break proteins down into smaller amino acids. Okay, now when trypsin is made, it actually is going to be kind of released in this inactive form, whereas you have a trypsin molecule and then an inhibitor that's attached to it. So it's released in an inactive form because if trypsin is released right away, it's going to start breaking down any proteins that it comes into contact with. And we don't want that to happen. We only want that to take place when we're actually inside the small intestine. So this enzyme is inhibited from the start. It isn't until this enzyme reaches the small intestine where another enzyme called enterokinase will come in and clip off the inhibitor, and we will then have an active trypsin molecule enzymes that's able to break down those proteins. Okay, and so it's very important to have inactive forms of enzymes in some situations because you do not want it to be active in the wrong spot. Another kind of mechanism of enzyme inhibition is actually how drugs work. So many drugs that we take are going to be inhibitors of enzymes. So for example, penicillin is going to be what we take when we get bacterial infections. And so what penicillin does is it is going to block an enzyme used to make the cell wall. So we haven't really talked about bacteria before in much detail, but bacteria have this cell wall that surrounds them that helps protect them. Okay, and so what penicillin will do is penicillin binding protein will come in and normally help make the cell wall for the protein or for the bacteria. But if we have this penicillin will come in here and block this enzyme that's really important for building the wall, and will make it so that the actual bacteria can't have cell walls, which makes them even more susceptible to our immune system. Now, ibuprofen or Advil also works in a very similar way. It's going to inhibit an enzyme active in sending pain signals. So when you have a headache, for example, and you take Advil or ibuprofen, what's happening is that you're preventing yourself from feeling the pain signals. The pain signals are still there, but the enzyme that's active in sending the pain signals is not functioning. And so you don't feel the pain anymore. Now, most of these inhibitors are reversible. So once the penicillin is no longer in the system or ibuprofen is gone, it'll stop and you'll go back to being able to send pain signals. But there are other things that are called irreversible inhibitors that once they bind, they will not unbind that enzyme. So nerve gas, for example, will bind to the active site of an enzyme that's really important in sending nerve impulses. And once it binds, it will not unbind. And that's really bad because if you can't send nerve impulses, you're going to die very quickly. Pesticides also work in this way. So we use the mechanism of pesticides binding to specific enzymes that are important, I think in neurotransmitters as well, so it kills whatever pest that we're trying to kill. The last thing I want to talk about inhibition of enzymes is this idea of feedback regulation. So there are many times in, for example, the human body that we are going to want to make some molecule. For example, in this case, we want to make some isoleucine, which is an amino acid. And the way that this takes place is that we'll have an enzyme that will help convert one amino acid, this one's threonine, and turn it into isoleucine. Now in this process, we take our substrate, threonine, and it attaches to this enzyme. There's multiple enzymes, but ultimately this is going to rearrange the chemical bonds in the structure and create this end product, isoleucine. Now, what we don't want to happen is turn all of our initial substrate, this threonine, into isoleucine. We want to make sure that we make isoleucine, but then we ultimately stop and, when we don't need any more. And so the way that this happens is sometimes the end product will actually be an inhibitor to this first enzyme. So in this case, what happens is that once the body has made up enough isoleucine, isoleucine will actually bind to the enzyme. And in this case, it's actually binding to a site, not the active site, but that's going to make it so that the active site will no longer bind this threonine on here, and it will stop this process. Okay, this allows for kind of the prevention of wasting resources, because you don't want to continuously do something if you've made enough. So in this case, what happens is that this will shut off the system once you have enough isoleucine. Okay, and we call this feedback regulation. This specifically is called negative feedback, where the end product is turning off the system. The last thing I want to talk about enzymes is that both temperature and pH are really important in enzyme function. So, for example, there are certain enzymes that will only work at a very specific temperature, okay? And they will only work at that temperature, and if they're at any higher temperature or lower temperature, they'll become non-functional. And this has to do with the structure. So at certain temperatures, the actual structure of the enzyme will start to have issues and sometimes will unfold and lose all of its kind of properties. So for example, there are certain human enzymes that are going to be kind of optimized at our body temperature. So most enzymes in the human body are going to be optimized for 37 degrees Celsius or 98.6 degrees Fahrenheit. So the reason why it's really dangerous to get a really high temperature is because your enzymes will start to be non-functional very quickly. So basically, if you get to, this one looks like 50 degrees Celsius, which I think is way too hot for any human, but you're going to start having issues. Now, there are other bacteria that are going to do with much higher temperatures, and that's for them. It's very specific to the organism. But for humans, most of the enzymes are going to work at that 37 degrees Celsius. Now, an interesting example where we have some enzymes in the human body that don't work at that temperature. So for example, the enzymes that are functional in producing sperm in males, they function at 93 degrees Fahrenheit, which is, quite a bit lower than 98.6. And so that is why the testicles hang outside the body, because they cannot be at that high of a temperature. They have to be at a lower temperature in order for sperm to be produced and be normal. And so that's a kind of a reason why, again, the testicles will not be within the main body cavity because the temperature is not right if it was inside. There also are going to be importances in pH. So pH, remember, we're talking about acids and bases. So certain pHs within our body, for example, our stomach enzymes are going to be very acidic. And so if you've ever thrown up, and I bet most people have at some point in their lives, usually it kind of burns your mouth and your throat because there's a lot of acids in your stomach. Okay, and so it helps break down the food, but when it gets into places it's not supposed to be, it can start to damage things. Okay, if we go to your intestine, it's going to be much more basic. Okay, and so the optimal pH for trypsin, for example, which is going to break down those proteins, it's going to be at a much higher pH than you would have for your stomach enzymes. Okay, and so if you have an enzyme that's made for the stomach and it goes into the small intestine, it's no longer going to be functional, and vice versa. So trypsin will not function in the stomach because it's not the correct pH. So pH is extremely important, as is temperature. Okay, and when you get outside of the normal pH or the outside normal temperature, your enzyme is no longer going to maintain its structure, which means it won't function correctly. We're now going to start talking about the membrane of the cell and how they regulate what goes into and out of the cell. So cells must regulate flow of materials to and from their environment. So what is on the outside of the cell versus what's on the inside of the cell is important to regulate. Okay, transport proteins are going to be one thing that we talk about now that are going to help move different substances across membranes. They can do it in two different ways. We're going to talk about passive transport when there's no energy required, and then active transport when energy is required. Okay, so for example, in this first situation shown in this image, we have this is a transport protein. It's going to require ATP to move these little blue molecules from one side of the membrane to the other. In this other situation right here, we're going to have some molecules that can move just directly across the membrane without any help. They don't even need a protein. And other ones are going to require a channel or protein that they can move across, but no energy is required. So passive transport is going to be over here where things can move from one side of the membrane to the other without any energy required. And on this other side, we're going to have some energy that's required, and it's always going to be ATP. So the membrane is quite complex. It's going to have, we talked about the lipid bilayer and how it's a fluid mosaic. So here we have the phosphoheads and we're going to have lipid tails, but we're also going to have lots of proteins that are embedded within the actual membrane. So some of these proteins are going to be channel proteins that allow for transport. Others are going to be self-signaling pathways. We also have enzymes that are embedded within the membrane themselves. We have some proteins that are going to help connect one cell to the other, and they're also going to have some cell recognition proteins on here. But it's quite complex, and our main focus today is going to be on this process right here, transport, and then we'll talk a little tiny bit about cell signaling at the very end. So let's talk about passive transport. This is going to involve something called diffusion. And diffusion is going to be movement of molecules across a membrane from a region of high concentration to a region of low concentration. Now, like we mentioned a lot earlier in this lecture, molecules have heat, okay? And that's going to cause them to vibrate and move and wander randomly. They do not just stay in one spot, they're going to move. Now, diffusion is going to be the tendency of these molecules of any type of substance to spread out into the available space. So instead of saying where everything is really tightly packed, molecules tend to spread out evenly. Okay, so if you do you can do this at home if you have some food dye. You take water and you put some food dye in here. What you're going to see over time is that the food dye initially is going to kind of all be very tightly in one spot, but over time it's going to diffuse, spread out evenly in the available space. And so this is what diffusion is looking like, and this is what's going to happen in our cells. So we're going to have diffusion of different molecules, and they're going to move and spread out in the available space. Now, passive transport is going to be the diffusion or the movement of a substance across a membrane without any energy being required. So a really good example of this is oxygen. So oxygen is going to be a molecule that is going to move directly across our membranes very easily. It does not require inner energy input. For example, when we breathe in oxygen, it ultimately is going to get into our bloodstream and then it needs to be delivered off to the cell. So the oxygen is just going to move right across the membrane without any energy being expended. Okay, it's very small and it can move through easily. Now, this term diffusion is going to be an example of passive transport. So diffusion is going to be moving molecules from one side to another. Okay, and so it's important to know that diffusion is a type of passive transport. It does not require any energy. Now, the definition of diffusion is the movement of molecules spreading out evenly into available space. So if we have here's our membrane, cell membrane, and we have the intracellular space and we have the extracellular space. So this means outside the cell, this is inside the cell. So membranes are going to allow some molecules to move directly past the membrane. For this example, we have all of these blue molecules on the outside of the cell and then on the inside. And so what's going to happen is over time, if these molecules are small enough, they will start to move across the membrane on their own and spread out. Okay, and at the end you get a point where you have kind of equal distribution of molecules on both sides. Now, why does this happen? Well, a substance will always diffuse down its concentration gradient. So this is a really high concentration. This has no concentration. So these blue molecules are going to move down their concentration gradient from where it's high concentration to low concentration until we reach what we call equilibrium at this endpoint. So here we can see this again. So here we have some molecules. These are red molecules, and they're small enough to move through this membrane. Now, the red molecules are going to be really high concentration over on this side, and there's none of them over here, so this is a very low concentration. Over time, what's going to happen is that molecules, because of their random motion, will start to move and spread out. Okay, and so during this first part, we're going to have net diffusion in this direction to the right, where there are less of the molecules. Now, the red molecules can move back and forth, but the net rate is going to be towards the right, where there's less of them. Okay, so things move down their concentration gradient. After a period of time, we will then get a point where we are at equilibrium. So molecules are still moving from one side of the membrane to the other, but there is no net one direction. It's basically equal going both directions, and therefore you have equal concentration of these red molecules on both sides of this membrane. So this is what's going to happen during diffusion. Now, some molecules can't move across the membrane on their own. They can move without the need of energy, but they're going to require these proteins that are going to help them move across the membrane. Usually this has to do with the chemical properties of the molecule. So for example, if we have a molecule that's really hydrophilic and loves water, it's going to have a tough time moving past these lipid tails, which are really hydrophobic, and so they tend to repel certain molecules. So instead of moving through the membrane directly, they're actually going to move through these channel proteins or these carrier proteins. And so in this situation, we are still going to be moving down the concentration gradient, and there's no energy required, but there are going to be proteins that are going to facilitate this. That's why we call it facilitated transport, where these yellow molecules are going to move down through this channel protein, down their concentration gradient, where there's a lot of yellow right here and very few of them down here, so they're going to move down this direction. And these purple triangles are also going to be moving through a carrier protein where from an area of high concentration to low concentration. And again, both of these do not require energy, so you see no ATP involved. So in this situation, we're seeing kind of passive transport of two types of molecules. So we have the blue molecules and we have the red molecules. They're going to be moving in different directions because they're both going to be moving down their concentration gradient, but ultimately we reach equilibrium where there's about even numbers of blue and red on both sides. So again, this is going to be diffusion. Now when we start talking about active transport, this is going to require energy. So if you're moving something against its concentration gradient, which means it's not the natural direction this molecule would move, it's going to require energy. Now, when you think of passive versus active transport, I want you to think of a river or a stream. So rivers and streams flow a certain direction. And so if you get in your inner tube and you're floating down the river or the stream, you're going to be going with the flow. And so you're kind of not having to expend any energy, you're just moving the direction that the water's taking you. That would be like passive transport. When we start talking about active transport, it would be as if you turned around in your inner tube and you start to try to swim against the current, moving up the river or the stream, which is going to require a lot of energy on your part because you're kind of going against the direction of the current, which is going to require you to actively swim that direction. So this is how active transport works. We are going to be moving molecules against the direction they normally would flow, and it's going to require energy in order for us to do that. So for example, if we look at this membrane, so here we have our membrane, and this is the outside of the cell, and this is the inside of the cell, And these are different ions. So we've talked about ions before. These are charged molecules. So this K stands for potassium, this Na is sodium, and this Ca2+ is going to be calcium. So what you're seeing is that our cells actively move potassium against its concentration gradient. Normally, potassium would tend to flow out of the cell because there's less potassium out here than there is inside, but our cells require us to have high potassium inside, and so we're going to actively pump it this direction. Sodium is the opposite, so there tends to be lots of sodium outside of the cell and not as much inside, but our cells have to create this gradient, and so they're going to pump actively sodium outside the cell. And that's the same thing that's going to happen with calcium. So calcium is tend to be kind of moved against its concentration outside of the cell. Okay, so active transport requires ATP. We're moving against the concentration gradient, which is why it requires that ATP. So you're moving kind of the direction against the current. So here we have an example of types of active transport. So you're going to notice that all of these have ATP involved with them. And in this case, we're moving these black molecules across into the cell. And you can see that the cell has higher levels of these black ones inside than it does outside. So the fact that we're moving this from outside to inside against its concentration gradient is going to require energy. So you're going to see ATP right here. When we move just one molecule, it's called a uniport, uni meaning one. So one molecule is moving this direction. We can have a different situation where we are moving two molecules, and in this case we're doing it in different directions. So we're moving the black molecules inside of the cell, which is going to require energy, and that same energy is going to help move these greenish gray squares outside of the cell against their concentration gradients. The last part that is shown on this diagram is the uniport coupled with the symport. So right here we have a channel that's actually not requiring any ATP, but what happens is it is dependent on this other channel that's using ATP. So basically it's moving these black molecules out of the cell against their concentration gradient, setting it up so then these black ones can run back through into the cell, which can help move these other squares. Okay, and so this one's kind of a complicated one, but the main thing to understand is that it does require ATP. So the next thing I want to talk about is osmosis. And osmosis is going to be the diffusion of water across the membrane. So if we're talking about water, instead of calling it diffusion, we tend to say osmosis instead. Now, before we start kind of getting into the details of osmosis, I want to define a few things for you. The first is that we have this term solute. Okay, solute is going to be the little molecules that are dissolved inside of the water. Okay, these molecules can be glucose, they can be salt, they can be lots of different things, but they're going to be these molecules that are dissolved within the water. Now, in certain situations, these solutes are unable to move from one side of the cell to the other. Okay, they are either going to be too big or there's something about them that makes it so the solute molecules cannot move. But what can move always is water. So water can move across the selectively permeable membrane at any point. So if you have lots of solute particles on one side and not very many solute particles on the other side, we can look at the difference between concentration of water versus the solute. So right here we have a very high concentration of water compared to solute, and over here we have more solute, so we would have a lower concentration of water. Now water is always going to move down its concentration gradient. Okay, so it's going to tend to move where there's less of it. So if you have more solute, that means that there's less water compared to the solute molecules. So water is going to tend to flow toward the direction of more solute. Okay, so it's moving down its concentration gradient. So if we're looking at kind of the amount of water per solute molecule, that's a really high concentration of water here and a very low concentration of water here. So water is going to tend to flow where there's less of it. So here we can see how this will work. So in this case, we are going to have molecules of sugar and we're going to have molecules of water. The sugar molecules are going to be in the purple and the water molecules are going to be in the blue. Now, when we look at these water molecules and these sugar molecules, we're going to say that the sugar cannot move, but the water can. So water can easily move from one side of the membrane to the other. So for some reason, in this case, the sugar molecules are just too big and they will not be able to flow. So you cannot move them down their concentration gradient. So instead of the solute particles moving, what's going to happen is water will move so that there's an equal concentration of water to solute in each side of this membrane. Okay, and so through the process of osmosis, water is going to tend to move down its concentration gradient. So where there's less of it, okay, there's less water on this side because we have more solutes. So water is going to move down into this side and is going to increase the amount of water so that we have a relative concentration of water to solute on both sides. Okay, so water is going to move to this side until there is an even number of water to solute. Now, it's going to be a little different because there are less solute particles on this side. So more water is going to move over here. So it looks like this has a higher water concentration, but if you compare it to the amount of solute, or these purples, it is going to be the same. So these would be at equilibrium. So here's another example of this, and we're going to start using some new terms that are going to describe the solution compared to kind of the cell. So on this side of this setup, we have it has water and it also has sugar molecules. And we have a selectively permeable membrane that allows for the water, but not these green sugar molecules to move. Okay, we consider this solution over here because it has lots of water and not very many solute molecules, hypotonic, which means less. Okay, so it has less of the solute molecules. On this side of the tube, we have lots of solutes, so these green sugars, and less water. So it's more concentrated, which means it's hypertonic. Hyper meaning more. So over time, what's going to happen is that the water does not want to have an unequal distribution of water compared to the solute. So what's going to happen is the water is going to move from a lower concentration of water, or from a higher concentration of water to the lower concentration. So there's less water over here. So water is going to diffuse into this other side, and over time, this is what's going to happen. So we then create something called an isotonic solution where both sides have the same amount of water compared to solute particles. So yes, this side has more water if we are going purely by volume, but if we go by concentration, these are absolutely identical. So if we compare the amount of solute to water, they are even on both sides. Now this slide has a lot of information on it, but this is going to kind of describe how we're going to talk about water and where it's going to move. So we have these three terms that we've already mentioned. We have isotonic, hypertonic, and hypo. Iso means same, hyper means more, hypo means less. So think of hypothermic, your temperature is too low. Okay? So an isotonic solution has equal concentrations of solute. So if we look at a cell, so here's our cell, and we're looking at the amount of solute particles compared to the amount of water. So on the outside of the cell, we are going to have 20% solute concentration and 80% water. Okay? Inside the cell, we have 20% solute and 80% water. Therefore, these are equal. So inside and outside of the cell are equal concentrations of water to solutes, and therefore water will move in and out of the cell at an even number. Okay, so there's no net direction. They're both going in and out at the same rate. Okay, so we call this no net movement of water. Okay, and that we basically are at equilibrium in this case. When we talk about hypertonic solution, the solution that the cell is in has more or higher solute concentration than inside of the cell. So if we look at our numbers, so outside the concentration has 40% solute concentration, which are these little molecules, and 60% water. If we look inside the cell, there's going to be 20% solute concentration and 80% water. So water is going to want to move where there's less of it. Okay, so if there's 80% water here and 60% water here, water is going to move out of the cell into the solution out here so that it evens out the amount of water on inside and outside of the cell. Okay, so hypertonic solution, a solution that has a higher solute concentration, so these are these green little things, okay, than another solution. So water particles will move out of the cell, and it actually causes a situation called crenation. Now, I'm going to show you red blood cells in a second. We do not want our red blood cells to look like this, where they're losing water and are shriveling. Okay, this is not a good situation. Our last situation is when we have a cell that is in a solution that has a lower solute concentration. So in this situation, the outside liquid is going to be 10% solute concentration, so it has only 2 of these little molecules, and it's going to be 90% water. Inside of the cell, it's going to be 80% water and 20% solute. So water always moves down its concentration gradient, so there's 90% water here and 80% water here, which means that water is going to move into the cell, causing the swell to actually get bigger in size and potentially can explode if there's too much water. So here's another depiction of that. Isotonic is the situation we want our cells to be in at most times. We want there to be kind of even numbers of the different solutes and water on both sides. So for example, in this case, we're looking at sodium chloride or table salt. You know, that's going to be something that is relevant to our bodies because we have salt that is going to be inside of our blood. So in this situation, the isotonic water is moving in and out at an even rate. When we have something called hypotonic, where we have more water on the outside and less water inside, water is going to move into the cell, which can then cause lysis, where the cell actually can explode if it gets too large. So if too much water is rushing into the cell, we can have a problem. Plasmolysis, or this is the situation with hypertonic, where instead what happens is there's more water inside the cell than there is outside, and water will leave, causing it to shrivel, and this thing called plasmolysis can happen, which is also not a good situation. So these two are not great. This is where we want to be. Now, water balance in plant cells is a little bit different from animal cells. So plant cells are going to have a cell wall around them, and the cell wall is going to help protect these cells from exploding if too much water rushes in. So those rigid cell walls will help create a barrier so that the cells do not explode if they're in a hypotonic environment. Now, plants tend to like to be in a hypotonic environment, which means that water is rushing into their cells, which makes their cells nice and plump, so that they are not shriveled. Okay, and so in plant cells, that this membrane or cell wall around the membrane is going to help prevent these from exploding. You can think of a cell without a cell wall as a water balloon, and if you keep on putting water in the water balloon, it'll eventually explode. But if you have the water balloon inside of a wooden box, the wooden box is going to help prevent too much water from coming in. So it allows for a lot, but at some point it's not going to let any more water in, and so it prevents it from exploding. Now, animal cells do not have a cell wall, and so if you have a person who drinks way too much water really fast, or if someone tries to inject water into your bloodstream, it will kill you. That is because our human cells will absorb water until they explode. So if you put this is a red blood cell, so a normal red blood cell needs to be in a isotonic solution where there's about the same amount of salt and water on both sides of the cell. But if you place a red blood cell in pure water, so you're putting it in pure water, water will rush into the cell and explode. Okay, so you basically the cell is no longer functional. If you put one of our red blood cells in very, very salty water, the water will leave the red blood cell and leave and go out into the solution, trying to equalize the amount of water on both sides of the cell. In that case, we do get a shriveled situation, so that's not good either. The plant cell, however, is going to have the cell wall that's going to help prevent it from exploding. So in the isotonic solution, water is coming in and out, equal levels on both sides. This is actually going to be not a perfect situation for a plant just because it's sort of flaccid. It prefers to be in a hypotonic solution where there's more water rushing into the cell, but the cell wall is preventing the cell from exploding. So this is the good situation right here. And then if you have a plant and you put the plant in salty water, all the water will leave the cell and will cause this situation right here, which is not good for the plant. And this is called plasmolysis right here. So in a plant, hypotonic solutions are the preferred solution, and in animals, we have to have isotonic so that we don't have our cells exploding or shriveling. And it all has to do with that presence of that cell wall. So the cell wall allows the plants to deal with more water without exploding than an animal cell. So here's what happens when a plant is given not enough water. It's going to basically shrivel. So here's a nice happy plant. And if you don't water this plant as often as it should, what's going to happen is that the water will start to leave the plant cells and go out into kind of the extracellular fluid, which then cause something called plasmolysis. Okay, so this is not a good situation for the plant, and plasmolysis can kill a plant if it's severe enough. So here's a summary of all the different aspects of transport that we've talked about. Remember that we have this first category, which is passive transport, which is going to move solutes from one side of the membrane to the other down their concentration gradient. This does not require any energy. When we're talking about water specifically, we call it osmosis. Otherwise, we can call it just diffusion when we're talking about the solute molecules moving against their or down their concentration gradient. And then we have active transport over here, which is going to require energy, which is going to be the movement of solute particles from a low concentration to a high concentration, which is not the natural direction things tend to move. So we have to use energy in order to pump these against their concentration gradients. Now, a little bit about how we move other larger molecules across our membranes. So some things are too large. It's going to be too hard for us to go through a channel protein. And so we have two different processes. One is called exocytosis and one is called endocytosis. And this is how we move very large molecules across a membrane. So exocytosis is going to be when these large molecules are going to be exiting or leaving the cell. Okay, and so we have these vesicles that are just membrane-bound vesicles that are going to contain some large molecules. They will actually fuse with the outside of the membrane and release the large molecules out into the extracellular fluid. Okay, so this is what this would look like. So here we have a membrane-bound vesicle, or just a vesicle is basically a storage container. We have these purple molecules, proteins maybe, something that needs to be leaving the cell. The cell membrane will fuse with the plasma membrane on the outside and will then release all of the molecules out into the outside of the cell. So this would be exocytosis. Now, the opposite of this is when cells can take in large quantities of things, but into the cell, so not leaving. So this is endocytosis, when they're taking things into the cell. So in this case, you can see right here, there are these purple molecules, and what's happening is that the cell is actually kind of engulfing and is going to close these molecules within its membrane and transport them into the cell. So this is what that would look like. So we have the molecules out here. They're going to be surrounded by the plasma membrane. The plasma membrane basically kind of comes and invaginates and is going to create this vesicle that then can move inside of the cell. Now, there are three different types of endocytosis. We have something called phagocytosis. The word phage means to eat, so this is cellular eating. So this is when you would have in this case, we have a cell that is going to be engulfing whatever this red molecule is. Okay, so it's going to engulf that and bring it into its body. We also have something called pinocytosis, which is cellular drinking. So there are times when cells will actually want to take in gulps of fluid into the cell. Okay, and so these blue regions, lighter blue regions, are going to be kind of the cellular drinking, or the gulps of the droplets into the cell. And the last type is something called receptor-mediated endocytosis. And this is when we have a kind of a very specific molecule that is going to be on the outside of the cell, and when a specific molecule comes in contact with this, and this one looks like a little heart. This is calthrin, and its coated pits is responsible for receptor-mediated uptake of extracellular molecules such as iron and growth factors. Okay, and so this calthrin molecule is going to be a kind of a tag that some of these molecules will then attach to, and they will be engulfed after they touch that receptor. Okay. The last thing I wanted to mention is the role of membranes in cell signaling. So cells are going to need to communicate with each other. They're going to need to be able to respond to environmental changes. And therefore, the outside of the cell is going to contain a number of different proteins and carbohydrates and lipids that are going to be able to interact with each other to respond to different stimuli. So this is actually showing a lot of different cell signaling pathways. For example, when nutrients, hormones, or stress come in, or this is calcium, this is chlorine. Stress can cause cell signaling pathways, microbial toxins, UV, and other stresses. Okay, they're going to then cause things to happen within the cell, so different pathways can be activated. And some of them will actually cause DNA to be read and new proteins to be made. In some cases, they will even trigger cells to die, which we call apoptosis. So the plasma membrane is going to help cells communicate with each other and also with their environment. So in this case, we have two cells, and they have chemical messengers these are just chemicals that are going to then combine to something on the plasma membrane and cause a signal pathway to go. In this case, it looks like it's going to the nucleus. So let's talk a little bit about how this communication will take place. So on the membrane of the cell, we're going to have these proteins, which we can call receptors. And the receptors are going to be able to respond to certain chemical signals. Now, we sometimes call these chemical signals ligands, and this ligand will actually bind to the active site of this protein on the membrane and will trigger something called a signal transduction pathway. And so this initial signal will ultimately cause a lot of different things to happen within the cell and will cause a cellular response. Now, you do not need to know what any of this in here means or what any of these things are, but the main thing to understand is that they can create and kind of relay a signal. So here's the signal. So it's kind of a message coming in. This protein then will start this cascade of events that ultimately, in this case, can cause a cellular response. And so what that cellular response is going to depend on the cell and on the signal, but it will convert kind of this signal into kind of chemical forms that can function within the cell. So this is an example of different cell signaling pathways and signal transduction. They often are extremely complicated. This, for example, is going to be a mechanism to get the immune system to start to respond. And so you can have these different ligands come in that are going to then cause these signal transduction pathways. And so obviously this is extremely complicated. You will not need to know anything about this, but it's just important to know that it is, there's lots of proteins involved, enzymes involved, but all of them are going to ultimately cause some response of the cell. And so in this case, the immune response. Now, cell signaling is going to be how cells communicate with their environment. It happens because we have receptor proteins on the outside of the cell. They receive a stimulus for a specific molecule. So some molecule is going to come in and kind of cause this receptor to respond. The stimulus then will trigger a chain of reaction inside of the cell, which will ultimately lead to some response. Okay, so the signal transduction pathway relays a signal that converts it to a chemical form that functions within the cell. Okay, and so I'm going to give you an example right now of how this happens. So let's just say you're out in the wild and all of a sudden there's a bear attack. Now this is a really cute bear attack, but let's imagine this was a real polar bear. You're on your own and it was actually coming at you. So what is your body going to do when this happens? So you're going to start doing something called the fight or flight response, where your body is going to prepare you to either defend yourself, you're going to fight the polar bear, or are you going to run away from the polar bear? Now, both of these things are going to require a lot of energy, and so our body is going to need to be preparing for a high expenditure of energy. Okay, you probably all have gone through this fight or flight response, not with a polar bear, but in other situations. Something scares you, or you get really nervous, and you start to feel your heart speed up, the beating, you breathe faster, you start sweating, all these different things start to happen. And so this is going to be a result of a cell signaling pathway that we're going to talk about. So this is what would happen if you actually came into contact with a real bear out in the environment. So we have a chemical messenger, which is called epinephrine. It's also called adrenaline. And this is going to be a chemical messenger released by the adrenal glands. The adrenal glands are these two little glands that sit right on top of your kidneys. So the epinephrine is going to be released into the bloodstream. It will then reach your muscle cells, and your muscle cells will have a receptor protein on the outside of their cell membrane. That protein will then attach to the epinephrine, which will then cause a signal transduction pathway. So lots of different proteins are going to be involved in this. There can be enzymes, different chemical reactions. Ultimately, what this is triggering is that your body is going to take glycogen that you've stored and is going to break it down into glucose so that glucose can be used for kind of you running away or for you fighting. So you're going to need glucose to power your muscles. Okay, so this is going to be the same pathway again as your fight or flight response. So this is what would be happening. And this doesn't necessarily have to have, you don't have to have an actual bear scaring you, but you can trigger this response and you're basically making it so that you're ready to fight or run away, which requires a lot of energy. Okay, so again, we have a chemical messenger, we have a protein receptor. This is going to cause a chain of events called signal transduction, where we ultimately will have a response, and in this case, we're going to break down glycogen into glucose. So that's going to be the end of the lecture. Remember, if you have any questions about any of this material, please email me and we can go over it. I will also go over this material in lecture review sessions, but please reach out if you're having any trouble. All right, thank you.